I would like to bring to the stage now Ann Paduri, KCNQ2 friend and champion Ann Paduri. Please, warm welcome for getting on that plane and coming to see our families from Harvard Medical School and Boston Children's.
Good morning. Thank you, Sarah. And if the plane wasn't going to take off, my dear husband was going to drive me down. It's wonderful to be here. Great to see so many friends and familiar faces and to meet new families who are here. I asked Sarah, do you really want me to talk about the basics? You all know so much. And she did say, it never hurts to go over the basics, and there are some new families. But you're new families in a new era. And 10 years ago, there wasn't talk of ASOs the way there is now. There wasn't earlier diagnosis. And so I think your new family's in an era of an information explosion. So I am going to take us back a little bit to some of the basics. Can I move this? Maybe. And also just reflect a little bit on the last few years and also where we're going. So after some basics, I do want to just give some lessons learned from some registry work we've done. We're hoping to now partner with Dennis, who's here, and who's going to take us forward. And then just in very, very broad strokes, talk about some clinical trial considerations and also a few comments about where we are in this moment in 2025 and how we really need to all come together to move forward. So what is KCNQ2? You've all had this wonderful balloon art up here all day. I was really, really, really impressed when I heard about, where's the balloon artist? There you are. Nice to see you. This might be all you need actually for the basics, but I'm actually going to go through in a few different forms. So what is this colorful curly Q structure? This is a representation of the KCNQ2 protein, and you might be thinking, what's she doing with ribbons and confetti? It looks like those little phone things when you're sending a little happy message. This is a rendition of the structure of how some of the protein structures actually will curl around and so on. So that's one way to look at the protein. Here's another way that looks a little bit more like this balloon that sort of marks out that part there is what's called the voltage sensing domain. It actually takes what we might think of as the electrochemical temperature of the cell. And what I want you to imagine is that down here is inside of a cell and up there is outside of a cell, and each of our cells in our body, including our brain cells, have to not only function on their own, but interact with the environment around them. And part of what this protein does is that it takes the temperature, it senses the voltage, the electrical potential of the cell. So that's what that part does. And then you've got the pore, which is over here, that when it opens up, lets potassium actually flow through. This is another picture, and I'm putting up, this is actually from a review article, and I'll put up some other pictures in part to show you that these are all the same thing. So if you are looking at the medical literature, you're looking at a scientific paper, and you're like, is it the ribbons, or is it the sticks, or is it the blobs, or is it the hot dogs? Like it's all of these things. These are just different ways of conceptualizing what this protein looks like. So here's the picture with the hot dogs that shows you that there's stuff on the inside, there's stuff on the outside, and it can open up and let potassium flow through. And then it can take different forms. A protein can twist and turn and do different things within the membrane. So I thought it might be nice just to go through a couple different pictures, and also just to remind you what the letters stand for. So K is a symbol for potassium. CN stands for channel, and there are lots of different proteins that look almost just the same, but Q2 is one example, Q3 is another example. These are proteins, are building blocks that sit in the membrane of the cell. So imagine a balloon, some visuals, you can imagine lots of balloons, but imagine just your cell is a balloon, and there's the salts on the inside and the salts on the outside, and they have to maintain a balance, otherwise they would either flow all of the salts out, or they would all flow in and the cell would pop, right? So there's got to be a balance all the time. So you have these different subunits, these different Legos or building blocks that are sitting there, taking the temperature, letting salts flow in and out. That's basically what KCNQ2 is. It doesn't do it alone. So I want some volunteers maybe to help. Maybe the four of you can help us. So KCNQ2 has a partner, which is called KCNQ3. It's very similar. So if you both would just put the number 2 up, you're going to be the KCNQ2 subunits. And then you two put the number 3 up. You're going to be the KCNQ3 subunits. And maybe get up and stretch. And we're going to pretend that the table is inside the cell and the ceiling is outside the cell. And if you can reach your hands together, that's a potassium channel heteromer, 4 pieces together. And right now, the potassium can't flow out because it's sort of configured in such a way. Now throw your hands up. And then it changes its conformation and it lets the potassium flow out. Thank you. That's an oversimplification, but it's how these different proteins kind of partner, and sitting in that little membrane of the cell, this is what happens. These proteins kind of aggregate. How they get formed, how they know to get there, how do they find each other, these are some of the sort of mysteries of biology I'm not going to go through today, but this all works in a coordinated fashion. And remember, these are 2 genes of about 20,000 or so genes that we have in our DNA. So this is one little tiny piece of the puzzle. But that's sort of what's going on, and this is critically important to what's going on with KCNQ2 DEE. So I'm going to take you back to high school biology. Anybody remember high school biology? So hopefully most people remember a little bit. What was in the books when I was taking high school biology was this very simple central dogma of molecular biology, that you have DNA, which is a double-stranded set of base pairs, A, T, C, and G, adenine, thymine, guanine, and cytosine. And then that DNA gets transcribed into RNA. You can sort of think of a transcriptionist copying it over. It's not an exact one-to-one, and suddenly you've got all these single-stranded pieces of RNA, and that's our blueprint that gets basically there's transcription that gets made into protein. Sort of shown another way, and that this is not KCNQ2, this is sort of a random sequence, and it's the nicest figure I can find, but they actually messed up the amino acids. So Aubrey, I'm looking at you, I know it's not a methionine. That's the bottom there. But the DNA, all these base pairs, A, T, C's, and G's, again, gets transcribed into RNA and translated into protein. And what happens when you have your mRNA, your RNA, your messenger RNA, is it gets put into triplets, this little triplet code. So you take those first 3, and that gives you the code for an amino acid. And then the next 3, that gives you a code for another amino acid. And then the next 3, and so on and so forth. How does it know where to stop? How does it know where to start? There's all sorts of signals around there. So this central dogma is a good place to start. It's where I want to leave you in terms of what's actually happening. It turns out that it's far more complicated. There's things upstream and downstream and in between, and coding DNA and non-coding DNA that now is understood in a way that we didn't understand when many of us were in school. But all of that is contributing to some of the nuance that you see, which is why when we saw these beautiful pictures of all of these beautiful children, they didn't all look the same. Their facial structures didn't all look the same. Their seizures may not all have looked the same. There's a lot going on within and around the gene, and then in all of those other 20,000 genes that might be influencing what's happening in the children. So what is it? It's all of these things. And this is just another cartoon from our Ion Channel blog from Ingo Helbig, just basically, again, going over that it really helps the cell tell us its level of electrical potential, or how excitable is it. And there needs to be a balance. And in all of our brains, there's normal electrical activity. People say, well, the EEG is measuring electrical activity, and people will often say, oh, is that bad? I'm like, no, we all have normal electrical activity, and there should be some balance in that electrical activity. And when there's an imbalance, very simply speaking, you can end up with seizures, and if there's abnormal circuits developing in the brain, you can have abnormal development and developmental disabilities, autism spectrum disorder, movement disorders, and so on and so forth. So this is sort of the core. I hope you've seen some of this in the last couple of days, and this is a review, but I think this is sort of the simplest way that I think it's nice to sort of look at, here the different parts, and you can have variants, as you well know, in any part of the protein, and they may have different effects depending on where they are. So what does it do? I mentioned it takes the electrical temperature, and that has to do with the cell by itself, but cells also have to talk to each other. Our neurons have to talk to each other at what's called the synapse, and I just wanted to show you where KCNQ2 fits in here. You can see it's marked right here. There's also sodium channels, calcium channels, lots of other channel genes and proteins, and that top cell is talking to the bottom cell. But they're all interacting, and when Sarah mentioned getting together with Danny and Danielle with STXB1 and their family, and SCN2A, which isn't cartooned here, but is also here, these are all proteins that are encoded by genes that are involved in these processes of keeping the cell at the right electrical potential and stabilizing it. And when that is not happening properly all the time, that's when we end up having the developmental and epileptic encephalopathies that we see. So it's good that they all got together. I think it's good that you get together for KCNQ2, but it's also good to think like the Australians have in GETA, that we're all not just on the same team in terms of being in this together as a community, but we're all on the same team in terms of the biology. We're sort of thinking about the same space. The scientists, Andreas, Al, the others you've heard from, are thinking about that this is what's in their head when they're thinking about these channels and proteins and what we need to modulate to try to rectify these abnormalities. I've just put this cartoon up. Anybody learn about action potentials at some point in life or education? Maybe a few. Again, I wanted to put some pictures on here so you can get a sense of what's going through our minds if this is how we studied. If anybody who'd studied neuroscience, which wasn't even a formal discipline back in the 90s when I was in college, we had to sort of go renegade and do this side thing when we were doing a biology major, but now it's a formal discipline. This is what we learn, right? We learn that there's a current, there's a resting potential, that minus 70 down there is in millivolts, and that's the resting potential, sort of the electrochemical temperature of the cell. And then stuff goes in, stuff goes out. And so for the potassium channel currents we're actually talking about normally a certain amount of current that has to go out to sort of reset the cell's temperature every time there's an action potential. And your millions of neurons are firing now, and this is going on in all of them. And most of the time, even in the children with KCNQ2 DEE, that's actually happening. It's just once in a while there's that, if our volunteers were to sort of not necessarily do that in coordination all of the time, and sometimes it wasn't quite right, that's when you have an abnormal instability, and that's when things don't develop properly. You have seizures and you have everything else. So again, lots of different ways of looking at exactly the same protein, but I also just wanted to give you a sense of like, well, what's in our heads when we think of these things? It's sort of all of these different things. So as you know, we have many patients sort of converging on this one protein. This is a few years ago, figure from Anne Berg's work with Sanal Mahida, whom some of you may have met the last couple of days from our group. And we just mapped on, as our artist has done here with the balloons, we've mapped on some of the variants, and also made a point that some of them that are in the darker circles actually happen more than once. There's recurrent variants. Now, how does that happen? Is there something special about those spots, or does every spot randomly get equal opportunity to have a variant? Every spot does have equal opportunity to get a variant. I sort of picture, imagine each of these different amino acids is a house, and it's raining. Which house is going to get the most raindrops? Well, if all the houses are exactly the same size, they're most likely to be hit statistically. But we're more likely to see symptoms maybe with certain spots, and there might be some that just give us more of an effect. So we're seeing them in people who come to the hospital for different conditions, or who go to the developmental clinic for different conditions in more abundance. Eventually, are there going to be more dots? I think Dennis will show you. Every single dot is covered, but they're not all represented here with KCNQ2 DEE. Some of the parts of this protein are more susceptible to actually leading to disruptions, and other parts you can probably disrupt and not necessarily see any abnormalities. So that's just sort of another way to look at this. Oops, I'm stuck here. All right. Dennis is going to update this for us, but this is just another way of saying if you show how the protein might fold, Dennis, I pinched this from you a few years ago, and you look at the blue dots and you look at the red dots, you might say, well, how can all of those people, each of them have a different variant in this gene, how can they all have the same thing? And they don't. The people who have the blue dots have dots or amino acids in part of the protein that are affected, that are on the sort of outside of the pore, so the backs of the KCNQ2 or KCNQ3. But if it was your backs that were affected, you all at my table here, and you were sort of putting your hands together and your back had something maybe a little bit off, like maybe forgot some sunscreen, had a little sunburn in one spot, so that's not going to affect your arm, right? So people who have the back part of the protein in this structure are fine, they're walking around and they're not actually having any issues, whereas if you had your arms affected, that's the red dots, all of a sudden things aren't going to work quite so well with your potassium flow. So it really does matter where on the protein. So it might look like every single spot is affected because the balloons that are appropriately shown in larger size and form and colored here are these sort of hot spots, the sort of really action ends of the protein. So I want to switch gears a little bit because I think this is the basics too. There's the basics of what do we learn in biology and chemistry that are in our heads when we're seeing our patients or when we're in the lab, but there's also the basics and the grounding that we all get from talking to you. And this is work that, where's Jim? I have to account to Jim every year and why this isn't published yet. So this is my sort of, we're still working on its slide, but we did interview a lot of families, and Brandon Obi, who is a wonderful pre-medical research assistant, worked with us with Sanal and interviewed some of you, including at one of the summits. And you know, the journal just didn't love how we put it together, and Brandon's in med school in New York City and doing great, and we're sort of trying to figure out how to get this into a more public format. It may end up being in the next Cambridge Elements, but we asked people, you know, that people are doing a lot of amazing science, right? You heard from Andrea's, Tim Yu's group, Aubrey's here, where we're seeing amazing science in the labs, and we hope it sort of will come and meet all of you in the clinic. But we asked people, you know, what do you hope to have from being part of a study, or even being from part of the community? And people said, we want to get a better understanding of how the gene works, understand how it impacts people with different variants. One parent said, because my son is now 21, and when he was born, they didn't even have any knowledge, and so now moving forward, we really want to get the information to learn about the different syndromes. One parent said that their child was diagnosed when he was 27, and they want to help future families, the new families. And I want to take a second here to talk about how these voices, it might have felt like they were qualitative interviews and it was just sort of the experiential aspect of things. And some of our colleagues, not the ones here, would say, well, you know, why does this matter? This isn't really driving the science forward. And I want to tell you that this is driving the science forward. These quotes, these sentiments, when we go to clinic now and we see a child at Boston Children's Hospital who has seizures that have started in the first year of life, within 1 to 2 days, we can get a hospital-supported research team there to enroll, if the family wants, in research, whether they're in the NICU or in the clinic. And then we can, at the same time, have a clinical genome test done. And within 7 to 10 days, if there's a diagnosis to be had, we'll have that diagnosis, and it can be given back to the family by the team, the genetic counselor, with a clinical report. And about half the time, we actually have an answer. And as you've seen in some of the signs around, when it's a baby, a young baby, the most common causes are 5 different genes, and KCNQ2 is one of the leading genes. This matters because it used to take us a year, 2 years, 5 years, 27 years to make those diagnoses. It's not easy when your kids are babies and you're getting this diagnosis and you look it up and it's not all peachy when you look at the literature. It really isn't, and we appreciate that. But we also are looking at families who waited decades to get these diagnoses when the technology just wasn't there. And we said, look, we've got the science, we have to bring it to the clinic. So I do want you to know that these interviews and these quotes really let us go to hospital leadership. They let us go to granting agencies and say that this is not a burden. This is what people are asking for. They need to know what their children have so they can try to do something about it. So what else have you told us? You've told us that there's all sorts of different seizure types that can happen. They can be convulsive, they can be myoclonic, they can be infantile spasms. And we said, well, great, let's figure out which parts of the protein give you which type of seizures. And you know what? We can't tell you. So we're not able to predict. So we can tell you what there is. You knew that already. You told us that, but it doesn't help us predict. If you look at the sequence and where the change is, it doesn't quite tell us. And also, the co-expressions. The term comorbidity was a thing several years ago when we were sort of laying out priorities for the field. It wasn't just the seizures. It is a lousy term. And if we had it to do over again, we would probably call it KCNQ2 neurodevelopmental syndrome with the following features, and you would tick off which of the following features might apply to your children. Sometimes it would be seizures, mostly it would be other things. And so what we did, and I won't go through this in great detail, but Anne Berg really put this together in a nice way to look at each of the variants we knew about and each of the children with those variants. What did they have, and did it matter whether they were still having seizures or not? And so if you had had recent seizures, that did predict that you were less likely to be independent, less likely to be walking, less likely to have independent motor function and communication. So seizures are important, and the drug companies I know also want to have a targetable, quantifiable measure, and the seizures in some cases early in this condition can be really important, and they do actually associate with other neurodevelopmental issues. So they're important, but they're only one part of the story. And again, I won't go through this, I'll just reference you to the paper, but really you can look at motor, you can look at communications, you can look at all sorts of things, and how long ago was your most recent seizure does predict in some ways the level of severity, but not always. So just they're interrelated, it's one brain, it's one protein that's disrupted. But in terms of day-to-day, it's not always the seizures that are really important. I do want to say, I saw Jim come in, I thought, there you are. We finally did publish our ezogabine experience. So our ezogabine experience, sadly, it's not available now, but we did see several kids who were getting ezogabine, and we had one child from Connecticut who really, really, really needed the ezogabine. It's been quite a saga that Heather Olson has taken them through with severe withdrawal, and we ended up getting an FDA IND to be able to use a product, and basically saw some improvements. I hope we'll see more compounds that are like this that won't have the side effect profile. I really hope that this what we learned from this experience was really, really important. We also learned that you could have really bad side effects from some of these drugs, and how you manage those side effects, urinary retention, needing to have catheterization, all those things was not fun, but maybe worth it if you had an improvement, but really not worth it if you didn't have an improvement. So we learned a lot. And then Sarah mentioned this is still anecdote, but this is the power of the moms. As Sarah mentioned, and two other families talked to us because Sarah put us in touch with them about exposure to what we sometimes call neuroleptic medications. These are medications typically used for behavior, and they're known as working on the dopamine system, or they're known on working on various neurotransmitter systems, but these are old, messy drugs. Some of the reason they might work is because they are old, messy drugs. They hit lots of different things at that synapse, not just one. And none of them are targeting KCNQ2, and I don't know that there's necessarily any evidence that they specifically are targeting individuals with KCNQ2, but we know that older kids with KCNQ2 who have the types of issues that Jackie has and that others have need help, and part of the help are the behavioral help. The t-shirt on that video is priceless. Severe behavior, I mean, it's like they knew there was going to be something going on where you were going to show us that. But the drugs sometimes work, but the drugs also bring about side effects, and the movement disorder side effects can be exacerbated in a condition where you have movement disorders. So we have 3 of these young people enrolled in the registry. We don't know if it's a coincidence or it's because KCNQ2 is in your movement center, the basal ganglia. And we need to look at this, and tardive dyskinesia could be something that is maybe more likely to happen in individuals with KCNQ2 DEE because that protein is in your movement center. So these are things that we're learning. And then the last thing I'll just say is that in terms of precision trials, there's so much excitement you're hearing about. Your surveys, your natural history, the data you're going to provide to Dennis when he puts all of these variants together and collects data that we've all been collecting and puts it in one place is really going to be important, because if we can stitch together the natural history for each child, but also in aggregate, we really can then say we know the mechanisms. Al George's lab and colleagues can study these in vitro in the dish, and we can really put together objective trial outcomes, whether it's seizure or non-seizure, quality of life measures that all need to be part of the trials that we do. We have all these pieces, and so it's just a matter of putting them together now. And then we really do need to think, as we're considering trials, is the model that somebody is describing in a dish, in an organoid, in a bunch of cells, in a mouse, is the thing that they're fixing with the ASO or the AAV or whatever it is, does that have relevance? So if they're saying, well, we're fixing the currents, well, that probably has relevance, but you really would like to see that they're fixing something that actually has meaning to the kids. And we're sort of trying to figure that out as we figure out precision trials. So we need good preclinical data, really robust data. We've got to be able to get to the right place at the right time in the brain, and maybe not just the brain. KCNQ2 is expressed in the gut. KCNQ2 is expressed in lots of different parts of the body. And so let's make sure we're thinking about all of those things, thinking about the potential benefits. Does the trial target meaningful outcomes? Potential risks? We're hearing now, very tragically, about some of the potential risks of brain-delivered AAVs, ASOs, and all those things. We have to be humble about the fact there's a lot that we are still learning, versus the risks of continuing standard of care, which may not be so great either. And so this is a real balance that you all will do. So how do we move forward? You all are an inspiration. And this is a slide that I put together for young physician scientists at UCSF and at the American Neurological Association and next week at the Child Neurology Society, because my colleagues, especially the young colleagues, said, look, we're kind of at a really, really tough moment as a scientific and medical enterprise in the country, where everything that our young scholars are doing and their livelihoods and their work is being really compromised by the lack of guarantee of federal funding and jobs, which is a huge part of the partnership that we do with the clinicians and all of you. So I'm reminding them to take their cue from you, to remember the mission, which is to improve the lives of people with neurological disorders. That's the mission of the NIH and the NINDS, where I was for about a year. It's the mission of the hospital and my clinic and every single one of us here. The passion we have is going to come from different places. For you, it's your family. For us, it's the people we see who come to us for help, and we'd like to do something better than the darts at the dartboard pills that we've been doing for decades in epilepsy care and behavioral care. We all need to self-reflect. Every single one of the young scientists you meet has to think about, like, why am I doing this, and how long can I do this, and how can I do this, and how can I partner across a community to be able to keep moving forward? And then we need to have resilience. This is not the easiest time, particularly in the US, for science and medicine. We need together to talk to everybody that we know, to sort of let them know that it is our mission and passion and our ability to reflect and say, yeah, we are doing this and we're doing this together, that keep us resilient. And that really gets to the community and why we're all here. I'm not going to go through every bubble, but this is another way of depicting what this community has done. This is from work that Al and Sarah Vekhuizen and Sarah James from Interviews has done through the elements. Look at this timeline back to the 60s when people actually were describing familial neonatal convulsions that turned out to be related to KCNQ2, all the way to now when we have databases and we have molecular diagnostics and we have ability to get diagnosis to family in a week or 2 weeks, and possibly treatments that are specific to KCNQ2 the next day. This is a lot of progress. And if we were sitting there in the 60s and thinking, okay, we're going to figure this out and we're going to have a treatment, we probably would have hoped it would have happened by now. Just to be honest, I think we're a little bit slower than we'd like to be, or a lot slower than we'd like to be in some cases. But it is moving, and what's moving it is all of you together with us. So I'll just highlight Brandon and Sanal, who've really been our team KCNQ2 at home in Boston. And I really, really want to thank everybody who's here. You inspire us and you keep things going. I can go back to these communities of young physician scientists, neurologists who are wringing their hands about, I don't know if I can pay the bills, and I don't know if I can do this, and what if I don't get my grant? And I can go and say, you know what? It's not really a choice. You're here doing it because you're committed. You have the mission and the passion, and you're going to look at your patients in the eye when they come to clinic, and you're going to tell them you're doing everything you can, and that comes from you. So thank you.